Well, good afternoon, everybody. Thank you so much for being here for the Cancer Center Symposium series. I'm Chris Lin, one of the GI medical oncologists here. I have the distinct pleasure of introducing Dr. Jun Yu from Johns Hopkins University to talk to us about pancreatic cancer. Dr. Yu graduated from Gannan Medical University in China, where he also completed his internship and residency in general surgery. He then completed his PhD at Kyushu University in Japan. He started his postdoctoral research fellowship in 2010 in Dr. Michael Goggins' lab in the Path Department at Johns Hopkins University, and then joined the faculty within the Department of Surgery at Johns Hopkins in 2017. From there, he had a rapid rise through the faculty ranks from instructor all the way to associate professor in 2019. He currently serves as the chief scientist in the Division of Surgical Oncology at Johns Hopkins. Dr. Yu has over 60 peer-reviewed publications and has an incredible amount of both predoctoral and postdoctoral mentees, and there's an extensive list here. Dr. Yu also received the American Pancreatic Association Young Investigator Award, and it's also received in 2018 the Nikki Mitchell Excellence in Pancreatic Cancer Award. We are very, very pleased to have him present his talk about learning from precision medicine in pancreatic cancer at Johns Hopkins. So please join me in welcoming Dr. Yu.
Thank you, Dr. Liu, for the kind introduction and providing the opportunity for me to present here. And today I'm going to talk about some experience at Hopkins, Hope for the Hopeless, learning from the precision medicine practice at Hopkins. So I do not have any disclosure. So let's start with some numbers. This is published earlier this year. So when you see the number here, for this year, 2019, there will be more than 56,000 people were diagnosed with pancreatic cancer. And for the estimated death for the pancreatic cancer patient is about more than 45,000. And also, when we look at the 5-year survival rate, and here, when you look at for all stages, and the 5-year survival rate is still around 8 to 9%. And when you compare the cancer cause death from both male and female, in the past decades, the pancreatic cancer cause death is the 4th death in both male and female. And this is a lethal disease. And why is that? When we look at here, the number is going to tell us. So when you look at the number at the diagnosis, and is about 10% of the patient were diagnosed with pancreatic cancer. Early stage is localized, and it's less than 10%. And also in my previous publication, we extracted more than 13,000 patients from CS database from all stage of pancreatic cancer patients from 2004 to 2011. And we calculated from the stage 1 pancreatic cancer progress to stage 4 is only taking 14 months, which means the cancer of pancreatic, this disease is growing rapidly. So, no detectable at early stage. And let's look at the other way. So is the patients resectable? Can we do the resection just resect the disease? Back to the number again. So among localized and regional pancreatic cancer, about more than 40%, and only 20% of the patients were resectable. And which means the others, 80% of the patients are not resectable, which means the overall survival is terrible. And the second point is not resectable. And then when we look at the chemotherapy, systematic therapy, when you look at here, so surgery alone is going to cause the 66% of the patient live longer than 10 years. And also if you put like chemotherapy with surgery, and will plus 14% of the patient live longer. And when you compare the median overall survival for the surgery alone and chemotherapy, this, well, from the number, there's a significance though, but from the reality, it's only like about 2 months. So no efficient systematic therapy. In our previous publication, we also checked the pattern of the recurrence after the surgery, because even for a lot of the time, the patients after the surgery will recur, and with sometimes they are locally recurrence, and sometimes they are like systematic in the liver, in the lung, and also when we counted the number, it's more than 76% of the patients had systematic recurrence. And also we checked when the recurrence occurred, and we used the mathematic calculation way to calculate it. If you have the recurrence like a month, like within 1 year, the overall survival is way lower than the recurrence of more than 1 year. And then we define the recurrence cutoff as 1 year. We call, if you have a recurrence more than 1 year, we call early recurrence. If it's more than 1 year, that will be a more late recurrence. So the fourth point is recurrent early. So is there any hope, and can we do better to make the patients live longer? Let's do it point by point. So for the first, no detectable at early stage. So can we increase the detectability for early stage cancer patients? Before we go forward, and I would like to introduce some histology of pancreatic cancer. So for pancreatic cancer, as we know, pancreatic cancer also has precursor lesions. And for the precursor lesions, there are 2 major pathways. One is PAN-IN pathway, from early stage of PAN-1, PAN-2, and PAN-3. And those cells will develop pancreatic ductal adenocarcinoma. And also there's another pathway, it's called IPMN cystic pathway, will occur from low-grade or intermediate-grade IPMN, which is benign, to high-grade IPMN is malignant, or you can say carcinoma in situ, to invasive IPMN. And for the early detection, I would say if we can target the PAN-3 or the high-grade IPMN or the early stage of the pancreatic cancers, then let's look at the molecular biology of the precursors and also the cancers. As you can see here, this is normal duct lined beautifully, and when you see some PAN-1 has some papillary causing, and also PAN-2, the nucleus are located like up downside, and also for PAN-3, you will have some bud like infiltrated in the duct. And once you have the pancreatic cancer, invasive tumor cells is going to break the basement, and also is causing the invasiveness. And when you look at the mutation for this, for PAN-1 is always have PAN-1 lesions and always have KRAS mutation. And for PAN-2 is always have KRAS and also P16 mutation. And for PAN-3 is always have like additional having P53 and SMAP4 mutations. This is our previous publication in clinical cancer research. We did like microRNA array for all the pancreatic precursor lesions. We checked all the microRNA's levels, and we found one microRNA targeted is called microRNA 196B. It's only occurred in the late stage of the precursor, which means like PAN3, and also in the cancer lesion, and no expression in the early stage of the precursors, which we can target this like marker. If we can detect this overexpressed in the patient, maybe there's something happening, especially for PAN3 or like a precursor, late stage of the precursor or the cancer. And this is another like pathway for IPMN, for high-grade IPMN and also invasive IPMN. As you can see here, this lesion is high-grade IPMN, and also surrounding part is the invasive tumor cells. And we did next-generation sequencing and figured that and found the same KRAS mutation in both high-grade IPMN and cancer, invasive cancer tumor cells, and also in P53 and in both lesions. As I mentioned, like P53 and SMAP4 always occurred in the late stage of the precursors. So switch the gear for the early detection. We also, at Hopkins, we published a paper showing the combining circulating tumor DNA with CA19-9 can increase the early stage of the cancer's detectability. As you can see here, if we combined both lesion and both like signals for early stage, stage 1A, stage 1B, and the detectability percentage is increased. This is another way to do the early detection. So we used microRNA. It's called microRNA MIR 1219. We used this marker to check the serum expression to help if we can distinguish the pancreatic cancer and also healthy or disease controls. And even among the early small stage of the cancer, if you see this, the cancer tumor size is less than 2 centimeters, still showed extremely high expression of the MIR 1219 versus the others IPM, normal benign disease, neuroendocrine tumor, chronic pancreatitis, normal and healthy, and other disease controls. So plasma microRNA is also can be used to help early detection. And we also used cell-free tumor DNA. Here I'm saying cell-free tumor DNA is different with circulating tumor DNA because we collected the DNA from duodenum juice, and we isolated the DNA from the duodenum juice and checked the mutation for KRAS, P53, and genus, and the other 9 cancer genes. And we, as I can show here, as you see, the KRAS mutation for control expression, we also used the digital next generation sequencing technology that I developed, and that you can see the score for KRAS mutation in control group is extremely low. It's not completely zero because, as you can see, like PANU1 and PANU1 and PANU2 always like express KRAS mutation. So in the chronic pancreatitis, always have PANU1, PANU2 in the normal pancreas. So it doesn't surprise us if you see the KRAS mutation even in the control group. However, if you compare the expression quantitatively for IPMN and also pancreatic cancer in the duodenum juice, the KRAS expression has significantly increased in both IPMN and PDAC. Even here, we cannot distinguish IPMN case and also PDAC case just by KRAS because for IPMN usually have more than 50% of the IPMN showing KRAS mutation, and for pancreatic cancer has more than 90 to 95% of the patients having the KRAS mutation. So we were not able to distinguish this though. As I mentioned, P53 and SMAD4 always occurred in the high-grade IPMN or late stage of the pancreatic cancer, or in the invasive pancreatic cancer, or in the PDAC. So when you just use KRAS P53 mutation and SMAD4 mutation, and it's beautifully distinguished the control and IPMN from pancreatic cancer. And also we can put all together with other 3 genes, with other 6 genes with, for example, BRAF, I did not show here, BRAF genus and TGF-beta receptor 2, and so on. And also even when you put it all together, you still can distinguish the IPMN cyst and also control from the pancreatic cancer group. So CFT, cell-free tumor DNA also can be used to distinguish the disease and also for detection. This is another clinical data from Hopkins, collaborated with Dr. Marco De Caro here in chief of the search arm in University of Colorado. So we use mathematic way to calculate the main duct dilatation, what the minimum, the cutoff size for the main duct dilatation to distinguish the cyst from benign, low grade or intermediate grade IPMN to high grade to invasive IPMN. As you can see here, 5 to 7 millimeters is the cutoff to distinguish those IPMN. And switch the gear for the detection. We also, in our previous publication, we also checked the germline mutations for the sporadic pancreatic cancer. So, and here, why I'm doing the sporadic pancreatic cancer, that's because a lot of the time for familial pancreatic cancer, you will do the genetic analysis and also sequential, whether you have the inherited mutation, genes mutation. But for a lot of the time, the familial pancreatic cancer is only less than 20%. And the majority is still sporadic pancreatic cancer. So is there any way you can use the germline mutation to predict whether you have the high risk of having cancer even in a normal, a non-familial, pancreatic cancer history? So we checked more than 800 patients with sporadic pancreatic cancer and checked the the pancreatic cancer suspect genes with BRCA1, BRCA2, and ATM. And when you compare the truncating mutations for this disease and the prevalence for BRCA2 is about 1.4% compared to the normal 0.4% significantly, and also similar to ATM. So as we know, BRCA1, BRCA2, ATM are familial pancreatic cancer genes, and also which here we also showed a similar result even in the sporadic pancreatic cancer. And also we did another analysis for like pancreatitis genes, which is CPA1 and the CPB1. And we also found the deleterious mutations for CPA1 and the CPB1 are contributed with pancreatic cancer. So back to the first point, not detectable at early stage. We have been working on it. So next point, not resectable. Can we resect more patients? Obviously, in the past decade, underwent Dr. Shuley, and he also was from Johns Hopkins. He stayed at Johns Hopkins for more than 30 years. He developed the technique for advanced resection and also for vessel involvement. And also, so does Dr. DeCario here. They do a lot of arterial resection for the localized pancreatic cancer patient. And some are in China. I'm not listed here though, but also at Hopkins after Dr. Schulich here, and also we do a lot of advanced resection for pancreatic cancer patients as well. So above the technique, we also have the neoadjuvant therapy could potentially benefit for the patient with locally advanced patient. So in the PMDC seen at Hopkins, from 2013, excuse me, from 2013 to 2017, and as you can see here, locally advanced patient is more than 461 patients. And we make the decision which patients should get neoadjuvant therapy, which should not. And also then after the neoadjuvant therapy, 20% of the patient underwent resection. And still 80% but we are making progress to resect more patients after the neoadjuvant therapy. And when you compare the overall survival for the locally advanced patient and with resection, this is with resection after the neoadjuvant therapy, the median overall survival is 35 months. And without resection, it's still low and is better than late stage, but still is low, 16 months. And also we did similar analysis for borderline resectable pancreatic cancer patients. As you can see here, performed resection had significant better overall survival compared to those without resection. So for this point, better surgical technique plus neoadjuvant therapy made more patients resected with better outcomes. The third point, no efficient systematic therapies. Do we have more efficient drugs? Well, there's at Hopkins. I don't know about how many trials in University of Colorado, medical oncology department. And at Hopkins, we have 13 active trials and one pending trial. So we are doing our best and try as we can to treat the patients. So, and also for the fourth point, can we manage the recurrence? So why there is recurrence? Even for some patients with clean resection, with R0 resection, but still you have the recurrence. So the one reason we thought is circulating tumor cells left behind after the resection, and also circulating tumor DNA and other moleculars. So as you can see here, what we found is the circulating tumor cells you can see with mesenchymal type, mesenchymal marker pan-CK, and also the vimentin. And also, these two cells are tumor-initiated tumor cells. Like you also can say stem cell-like circulating tumor cells. And we published several papers for circulating tumor cells, and circulating tumor cells phenotype can predict recurrence and survival, and also CTC can express like tumor-initiating cells, and also circulating tumor cells like can be seen even in IPMN cyst pancreatic lesions. So, circulating tumor cells, yes, no, is not just one step. And also, we checked the circulating tumor cells in a longitudinal way and with different time point. As you can see here, this is a patient with recurrence. And this time point is the like OR after the surgery. And the yellow line is mesenchymal type of the circulating tumor cells. The blue line is the epithelial type of the circulating tumor cells. The black line is the total number. As you can see, after the surgery, because this time point is when before or like pre-incision. So we collected blood and also did circulating tumor cells isolation pre-incision of the surgery. So you can see the circulating tumor cells number are dropping during the treatment, and also after several hundred days, and you see the numbers is rising, and also this time point after the surgery, 472 days recurred. And actually, this recurrence was seen by the CT image, did not seen at this stage. However, we have the signal, like a rising signal, and of the CTC showed there's something happening like with this disease, and also the signal showed prior the like CT imaging, which means like CTC can be a better like monitor biomarker than CT imaging for recurrence detection. And this one is the patient did not recur, and we did not see any circulating tumor cells even after the several hundred days. And for circulating tumor DNA is another marker you can use. And here is just like an example I'm showing here is like we did see the circulating tumor DNA rising compared to CA19-9 because this patient did not have any like rising CA19-9 expression though, but we did see the signal of the circulating tumor cells, DNA, and this mark means the patient died. So we use CTC and ctDNA to monitor the recurrence. So back to the hope. Try to detect at early stage, try to resect more patients, try many systematic therapies, try to use biomarkers to monitor the recurrence. And additionally, I would add another one, precision medicine in pancreatic cancer. So I'm going to introduce the precision program at Hopkins. So usually we have a patient will be seen by the team in the PMDC group, and at the diagnosis, we will make the diagnosis first and confirmed by FNA. And FNA, we will take several like cause biopsies. So two biopsies here will be used for clinic purpose. And one will be used for the diagnosis, and one will be stored for future, like re-review. And also for additional cause, we will use for research purpose. And one will go to next generation sequencing, and one will go to 3D organelle culture in the lab, and one will be stored in the biobank for future analysis. And for 3D organelle culture, we will culture them and purify the tumor cells from the FNA biopsy. And also we will do pharmacotyping, not just limited to current commercialized regimens, chemotherapy. And also we will use this like generated cell line to co-culture with immune cells to do some basic science for immune therapy purpose. And for blood, we will collect the whole blood for circulating tumor cells isolation. And one part will go to next generation sequencing for genotyping. And one will do immunofluorescence staining for phenotyping. And also we will grow the circulating tumor cells, live cells in a 3D organelle culture system. And we will bank those CTCs. And also for plasma. So we will collect the plasma. One part is for NGS for circulating tumor DNA. And we also keep 1 to 2 vials for proteomics. And we will biobank them. And actually one part for that, we will analyze exosome from plasma as well. And for the procedure for the patient, we will do conventional therapy for like neoadjuvant therapy. And before and post neoadjuvant therapy, we will collect the blood. And similar to the previous one, we will collect all the informations here. And then we will do the surgery if the patient is resectable. And for the after the surgery, there are 2 parts. One is tumor, resected tumor, and one is blood. So for the tumor, so we separate that in 2 parts as well. One is for clinical purpose, for diagnosis, confirmation, and also tumor regression grading. And for research, we will do the next generation sequencing, and also we will use the resected specimen to do 3D organelle culture system, and also pharmacotyping. And we will biobank the rest like a specimen. Blood, similar. We will do the regular way and to keep like a plasma and also circulating tumor cells. And after the surgery, we will do the adjuvant therapy, and we will collect the blood pre-adjuvant therapy and post-adjuvant therapy. And also during the every 3 months follow-up, the patient will come to Hopkins, and we will collect the blood, and also we'll collect all the plasma and also circulating tumor cells. So here, so I'm showing here FNA as the diagnosis part, and why we did NGS is because we want to know the baseline of the sequencing stuff, and also we will use that to confirm here with tumor grading and also next generation sequencing here if they showed same or different like tumor cells or mutations. And one part we do that is so here like we use, and another way is prospectively you can use the information here. So you can do the NGS and check the profile and expression and use the tumor regression grading system. You will know if one marker can be used to predict the the response, chemo response. That's what we did in the preliminary data showing. So for the well to medium response of the after the neoadjuvant therapy, the genes, 31 genes showed different expression compared to the poor responded patients. And also from the other way, highly expressed genes only showed in the poor respond case. Here, you actually, in the like a prospective trial, you can use the gene expression at this stage. You can decide whether this patient should get neoadjuvant therapy or not, because not everybody will get the response from after the neoadjuvant therapy. So pretreatment NGS markers may help select patients for neoadjuvant therapy precisely. And also for the pharmacotyping, because we collect all the live cells before the treatment and also after the treatment, then we can know if the neoadjuvant therapy will shift the tumor survival after the treatment, and if they are exactly the same, the tumor cells all are different, which means which will guide you to give whether you have to give the different a regimen for adjuvant therapy setting, if you want to keep the same or you want to switch to the others. And also here we can, after we have the live cells, generated cell lines, we can also do some co-culture with immune cells to do some a basic science research for the immunotherapy. So that's the beauty of the precision medicine here, and also for international collaboration. So in the past, past year, so I was lucky to organize the conference. It's called Pancreatic 2018 with my boss, Dr. Chris Wolfgang, and it's more than 300 the international experts joined the conference for pancreatic cancer, and the beauty for this one, and also the future, the result. What we got is we built up the broad connection. So from Johns Hopkins with Europe, and that's and also we have a lot of connection with North America, including United States and also Canada. And also we have a lot of relationship built up based on that with Asia countries, from China and also from Korea and also from Japan. So besides the precision medicine program, so additionally, I'm going to talk one more project what we done, and this is the project my PhD candidate Haijia Hu did. So this is checking the germline mutation for the pancreatic cancer with a sporadic, and here, because it's still unpublished data, so I put X here as the mutation. So the left figure is the disease-free survival, and the right one is the overall survival. As you can see here, if you have a mutation in germline, and the overall survival is significantly better than wild type. And also, disease-free survival is significantly better. And when you compare the overall survival, and also it still has the significance. And also, this is for all cases, regardless what kind of treatment you get. So we then subgrouped the cases into the resection and the chemotherapy. So when you check the disease-free survival, so let me explain this. So if you have a mutation, and also you get the resection, and also get the chemoadjuvant therapy, the significance is still there, better than wild type. And also for overall survival, if you have the mutation after the resection and the adjuvant therapy, and the overall survival is significant better than wild type. Then we thought about, what about if you just have the resection only without adjuvant therapy? So this is the data for the mutation. So if you have a mutation and also with resection and no chemotherapy, there's the significance lost. There's no significance for both mutation and wild type. And also, when you check the overall survival, the resection with no chemotherapy, there's no significance either. So compare this figure and to this figure, which means the chemotherapy is driving the patients with mutation having better outcome. So this is additional analysis for the subgroup. So we compared the patients with mutation. Okay, so all this cohort is all patients have mutation. And then we had vaccine, which was developed by Liz Jeffrey at Johns Hopkins, treatment and resection and chemotherapy versus those other regimen for the neoadjuvant therapy with resection and chemotherapy. And as you can see, the blue line here, vaccine-based patients had dramatic, significant, better outcome compared to those, even those patients did not get vaccine therapy. Although all these patients were genes mutation carrier, indicate the vaccine worked for this patient having the mutation. So then we thought about, what about if the patient does not have the mutation and if you give the patient also the same vaccine neoadjuvant therapy, what does it happen? So we luckily, in the retrospective data collection, we had 2 patients did not having the mutation, wild type, and also had the vaccine treatment, and versus those vaccine-treated patient and with mutation. And when we look at the survival, both patients having wild type died about 16 months after the surgery, even after the adjuvant therapy. And for the patient having the mutation and also underwent vaccine treatment, and one patient died 43 months, one patient died 53 months, and one patient is still alive after the surgery and is 92 months. Actually, this patient was resected by Dr. Schulich. So, hope for the hopeless. This is actually, I was thinking about a picture. I cannot find that picture, but I saw a TV show. It's called a Prison Break, and also they use that wall is called the devil. And so here I use similar, but this is the gray board is a wall, the thick wall of the disease, the pancreatic cancer disease. So early detection, only one hole, is not going to break the wall. Only more resection is not going to break the wall either. And a better chemotherapy might be benefit for some patient, but it's still not be able to break the wall. And monitor the recurrence, yes, it will give you better outcome, but still it's not gonna solve the problem. So if you have all this together, and will be with the wall, the thick wall of the pancreatic cancer disease, I think will be broken. And this is the member of the pancreatic cancer program. Actually, that was me. And this picture was taken in 2000, I think it's 15. That's before I moved to surgery department. And Yes, and this is Dr. Liz Jaffe and Dan Hero here, and this is my current boss, Dr. Wolfgang, and this is my ex-boss, like Michael Goggins in the pathology department. Yeah, this is Yang Li here, and this is the current my group, and as you can see with my boss here, the chief surgical oncology, Dr. Wolfgang, and also with Liz Jaffe, Dan Lahiro, and pathologist Liz Thompson, and Lei Zhang, med oncologist, and also 4 other surgical attending here. And all these are my students and postdocs here. So thank you.
Thanks, Dr. Yu. We'll open it up for any questions for Dr. Yu.
I have a couple slides mentioned the vaccine stuff. Do you use the neoantigen as a vaccine target to treat the patient, or what's the vaccine target?
So that vaccine actually is not typical vaccine. It's like a boost. So it's causing the immune cells infiltrating in the tumor. So it's a boost, yes, which was developed by Liz Jaffe, I would say at least 15 to 20 years ago. Yeah, we're still using that, yeah.
Other questions? So I have a question regarding ctDNA kinetics.
Yes.
So if you have a patient that goes from an IPMN to cancer, have you guys been able to look longitudinally to see if circulating tumor DNA increases in allele frequency as you go from one to the other?
Yes, actually, I did not show that slide here, though, in my paper, this one, let me see. So in this paper, we also checked the longitudinally way. So actually, in this cohort, I would say this cohort, because some patient had IPMN and with p53 and SMAD4 mutation, and these cases were actually we detected p53 mutation, and 4 of them developed pancreatic cancer for after at least 18 months. And which show, I did not show here though, but we showed the signal is there and also detectable, and for high grade, at least a high grade of the dysplasia, then CT imaging, you can see the tumor and also cancers. And that's the beauty of using the cell-free tumor DNA technique to monitor or for the early detection of the pancreatic cancer. Yeah.
Other questions?
At Johns Hopkins, are you currently genotyping all of your patients that come into the clinic to see if the track are associated with some of the circulating microRNAs and circulating tumors, such as expression box?
Yes, we are.
And so is that helping in terms of your detecting which patients are going to develop recurrence and also resistance to certain chemotherapies?
For that analysis, we haven't done, but we just genotype those and also keep the record, and also for circulating tumor cells, because actually here you might be able to find the same mutation, but what else can tell you? So we want to know why the circulating tumor cells can escape the immune attack and also in the blood system, and that's why we are doing the culture for circulating tumor cells to do more analysis for that. But like I said, so current analysis is not able to distinguish what patients you will have the recurrence or you have more circulating tumor cells based on the genotyping at the first baseline point. Yeah.